Good morning to all. I am Dr.K.Amutha, Assistant Professor, Department of Zoology, A.D.M College for Women, Nagapattinam. Now I am going to talk about Golgi Apparatus. It is one of the cellular organelle. The Golgi Apparatus or the Golgi body or Golgi complex is simply Golgi. It is a cellular organelle present in most of the cells of the eukaryotic organisms. It is referred to as the manufacturing and the shipping center of the cell. Golgi is involved in the packaging of the protein molecules before they are sent to the destination. These organelles help in processing and packaging the macromolecules like proteins and lipids that are synthesized by the cell and hence act as the post office of the cell. It is otherwise known as the post office of the cell. Golgi apparatus was discovered in the year 1898 by an Italian biologist known Camillo Golgi. The diagram shows the Golgi apparatus. It includes the vesicles, tubules and cisternae. Structure of Golgi apparatus. Under the electron microscope, the Golgi apparatus is seen to be composed of stacks of flattened structures that contain numerous vesicles containing secretory granules. The Golgi apparatus is morphologically very similar in both plant and animal cells. However, it is extremely pleomorphic in some cell types. It appears compact and limited. In others, it is spread out and reticular, that is net-like structures. Typically, however, Golgi apparatus appears as a complex array of interconnecting tubules, vesicles and cisternae. Cisternae, it is the simplest unit of the Golgi apparatus, is a cisternae. Cisternae, its diameter is about 1 millimicron in diameter or central flattened structures. They are otherwise plate-like structure, saucer-like closed compartments that are held in parallel bundles or stacks one above the other. In each stack, cisternae are separated by a space of 20 to 30 nanometer, which may contain rod-like elements or fibers. In each stack of cisternae forms a dictyosome which may contain 5 to 6 cisternae in animal cells or 20 or more cisternae in plant cells. Comparatively, in animal cells it is less. Each cisternae is bounded by a smooth unit membrane, that is the thickness is about 7.5 nanometer, having a lumen varying in width from 500 to 1000 nanometer. The margins of each cisternae are gently curved so that the entire dictyosome of the Golgi apparatus takes on a bow-like appearance. The cisternae at the convex end of the dictyosome comprises proximal forming or cis face and cisternae at the concave end of the dictyosome. It comprises the distal, maturing or transphase. Next one is the tubules. A complex array of associated vesicles and anastomosing tubules, that is the thickness is about 30 to 50 nanometer in diameter, surround the dictyosome and radiate from it. In fact, the peripheral area of the dictyosome is fenestrated, that is lac-like in structure. Vesicles. The vesicles 60 nanometer in diameter are of three types. First one is the transitional vesicle, second one is the secretory vesicle. The transitional vesicle are small membrane limited vesicle which are thrown to form the blebs of the transitional endoplasmic reticulum to migrate and converge to cis face of Golgi, where they coalesce to form new cisternae. Second one is the secretory vesicles or varied size membrane limited vesicles that discharge from margins of cisternae of Golgi. They often occur between the maturing phase of Golgi and the plasma membrane. The third one is the Clathrin coated vesicles, or they are spherical protuberances about 50 millimicron in diameter and with a rough surface. They are found at the periphery of the organelle, usually at the ends of single tubules, and are morphologically quite distinct from the secretory vesicle. The Clathrin coated vesicles are known to play a role in intracellular traffic of membrane, so that the Golgi is known as the traffic police, and of secretory products that is between endoplasmic reticulum and Golgi, as well as between the GLR region and the endosomal and lysosomal compartments. Next one is the functions of Golgi apparatus. The first function is it is a vesicle or often referred to as the traffic police of the cell. They play a key role in sorting many of the cells, proteins and membrane constituents and in directing them to their proper destinations. Their main function is directing them to their proper destinations. To perform this function, the Golgi vesicles contain different sets of enzymes in different types of vesicles, that is the cis, middle and transcisternae, that react with and modify secreted proteins passing through the Golgi lumen or membrane proteins and glycoproteins that are transiently in the Golgi membranes as they are en route to their final destination. They attain their final destination through this channel. The Golgi apparatus hence acts as assembly factory, the second function. The Golgi apparatus hence acts as assembly factory of the cell where the raw materials are directed to the Golgi apparatus before being passed out from the cell. In animals, the Golgi apparatus is involved in the packaging and exocytosis of the following materials, that is the zymogen of exocrine pancreatic cells, the mucus, that is the glycoprotein, secretion by globular cells of the intestine. Lactoprotein, that is a casein, that is from milk, the secretion by mammary glands of the murocrine secretion of compounds, that is the thyroglobulins of thyroxine hormones by thyroid cells, secretion of tropocollagen and collagen, then formation of melanin granules and other pigments of skin, and formation of yolk and vitelline membrane of growing primary oocyte cells. Third function, it is also involved in the formation of certain cellular organelles such as plasma membrane, lysosomes, acrosome of spermatozoa and cortical granules of a variety of oocytes. The next one is fourth function. They are also involved in the transport of lipid molecules around the cell. The Golgi complex also plays an important role in the production of proteoglycans. The proteoglycans are molecules that are present in the extracellular matrix of the animal cells. Sixth function, it is also a major site of synthesis of carbohydrate. These carbohydrates includes the synthesis of glycosaminoglycans. Then the Golgi attaches to the polysaccharides, which then attaches to the protein produced in the endoplasmic reticulum to form the proteoglycans. That is in the rough endoplasmic reticulum. The Golgi involves in the sulfation process of certain molecules. The process of phosphorylation of molecules by the Golgi requires the import of ATP into the lumen of the Golgi. Next function is in plants, the Golgi apparatus is mainly involved in the secretion of materials of primary and secondary cell walls. Example, formation and export of glycoproteins, lipids, proteins and monomers of hemicellulose and cellulose, lignin, etc. Now let us see about the structure of nucleus. First, what is nucleus? The cell nucleus is a membrane bound structure that contains the cell's hereditary information and controls the cell's growth and reproduction. It is the command center of the eukaryotic cell and is commonly the most prominent organelle in a cell, accounting for about 10% of the cell's volume. In general, a eukaryotic cell has only one nucleus. However, some eukaryotic cells are eunucleated cells. That means without a nucleus. For example, red blood cells RBCs, whereas some of the multinucleate consists of 2 or more nuclei. For example, slime molds. The nucleus is separated from the rest of the cell or the cytoplasm by a nuclear membrane, as the nucleus regulates the integrity of genes and gene expression. It is also referred to as the control center of the cell. This is the diagram shows the structure of the nucleus. In the nuclear pore, nuclear membrane, then chromatin reticulum, the center part side having nucleolus, the nucleoplasm, and the nuclear membrane is otherwise known as nuclear envelope. The structure of a nucleus encompasses the nuclear membrane, nucleoplasm, chromosomes and nucleolus. First one is the nuclear membrane. The nuclear membrane is a double layered structure that encloses the contents of the nucleus. The outer layer of the membrane is connected to the endoplasmic reticulum. Like the cell membrane, the nuclear envelope consists of phospholipids that form a lipid bilayer. The envelope helps to maintain the shape of the nucleus and assists in regulating the flow of molecules into and out of the nucleus through nuclear pores. They use the nuclear pores for the import and export of the materials. That is the exchange of materials. The nucleus communicates with the remaining of the cell or cytoplasm through several openings called nuclear pores. Such nuclear pores are the sites for the exchange of large molecules such as proteins and RNA between the nucleus and the cytoplasm. This is the exchange of materials from the cytoplasmic region to the nucleus. A fluid filled with space or perinuclear space is present between the two layers of a nuclear membrane. Next one is the nucleoplasm. Nucleoplasm is the gelatinous substance within the nuclear envelope, also called the karyoplasm. This semi-aqueous material is similar to the cytoplasm and is composed mainly of water with dissolved salts, enzymes and organic molecules suspended within. The nucleus and chromosomes are surrounded by nucleoplasm, which functions to cushion and protect the contents of the nucleus. Nucleoplasm, this also supports the nucleus by helping to maintain it safe. Additionally, nucleoplasm provides a medium by which materials such as enzymes and nucleotides, that is DNA and RNA subunits, they can be transported throughout the nucleus. Then substances are exchanged between the cytoplasm and the nucleoplasm through the nucleopores. The next one is the nucleolus. Contained within the nucleus, it is a dense membraneless structure composed of RNA and proteins called the nucleolus. Some of the eukaryotic organisms have a nucleus that contains up to 4 nucleoli. The nucleolus contains nuclear organelles, which are parts of chromosomes with the genes of ribosome synthesis on them. The nucleolus helps to synthesize ribosomes by transcripting and assembling ribosomal RNA subunits. That is, there are 2 subunits in the ribosomes. Here they are assembled. These subunits are joined together to form a ribosome during protein synthesis. The nucleolus disappears when a cell undergoes cell division and is reformed after the completion of cell division. Chromosomes. The nucleus is the organelle that houses chromosomes. Chromosomes consist of DNA, which contains hereditary information and instructions for cell growth, development and reproduction. Chromosomes are present in the form of strings of DNA and histones. That is, the protein. Histones are the protein molecules called chromatin. When a cell is resting, that is not dividing, the chromosomes are organized into long entangled structures called chromatins. The chromatin is further classified into heterochromatins and echromatin based on the function. The former type is highly condensed, transcriptionally inactive form, most present adjacent to the nuclear membrane. On the other hand, euchromatin is a delicate, less condensed organization of chromatin, which is found abundantly in a transcribing cell. Functions of nucleus. The nucleus provides a site for genetic transcription that is segregated from the location of translation in the cytoplasm, allowing levels of gene regulation that are not available in prokaryotes, that is available only in eukaryotes. The main function of the cell nucleus is to control gene expression and mediate the replication of DNA during the cell cycle. It controls the hereditary characteristics of one organism. This organelle is also responsible for protein synthesis, cell division, growth and differentiation. Storage of hereditary material. The genes in the form of long and thin DNA, that is the ribonucleic acid strands, referred to as the chromatins. Here the storage of proteins and RNA in the nucleolus. The nucleus is a site for transcription in which messenger RNA that are produced for protein synthesis. During the cell division, chromatins are arranged into chromosomes in the nucleus. Production of ribosomes. Here the ribosomes are otherwise known as protein factories in the nucleolus. Selective transportation of self-regulatory factors and energy molecules through nuclear pores. Thank you.